Okay, this video I'm just going to run through a couple of DNA manipulation exam questions. So this one here, I'm just going to check if it's on the next page. It's on page 3 in your notes. So this says sickle cell anemia is a serious inherited blood condition. It leads to tiredness and kidney or heart failure. So without treatment, children usually die before the age of 10, and it's due to a change in the gene which codes for beta hemoglobin. So we did do a prac on this, and hopefully you can understand what we're talking about here. So there are 2 alleles for beta hemoglobin. HbA, is normal beta hemoglobin, and HbS is the changed or the sickle cell hemoglobin. So now what we can do is we can actually genetically test people to see whether or not they carry this HbS. And it uses PCR, restriction enzymes, and gel electrophoresis. So we're going to be combining all of those things together. This is the restriction enzyme, MST2. And what it does is it cuts the DNA like this, and it produces this. So what is the term that we use to describe the fragments? And hopefully you've had a chance to go through this. If you haven't, stop the video here, go and give this question a go, and then come back and read the answers or go through the answers with me. So hopefully you've come back. And the term used to describe this is going to be sticky ends. Yay! You can see the sticky ends there. They're poking out on either side. So molecular studies have shown that the sickle cell allele differs by only 1 base pair. We haven't done mutation yet, but we will come back to that. This base change occurs in a 7-base sequence that is recognized by that restriction enzyme. So essentially what's happened is this bit here has switched around and become this mutation. So the PCR products, so that means these guys here, have been gone through PCR, and now we're putting this MST2 into the mixture and we're digesting them. And then we're going to run that through the gel electrophoresis and see how we go. So how is the action of this enzyme going to be affected by the mutation? So you guys can see this a bit easier. If I change this one here, will the restriction enzyme bond? The thing is no, because what you've done is you've changed your substrate. You've changed the sequence that it's looking for. So therefore, you get no cutting. So what we should end up with, have I got answers in this here? Here we go. What we should end up with is the enzyme does not cut the DNA, or the enzyme no longer recognizes the sequence. So no cut or no recognition of MST2 sequence. It's a very rough thing. All right, so now we're going to look at this and we're going to mark on the gel what banding patterns you would see. Now, this is a great question, but it involves a little bit of revision on what we mean by alleles and what we mean by genes. So essentially remember that you get 2 copies of every gene. You get a copy from mum and a copy from dad. So this one here, we're going to say that one's from mum, and this one here, we're going to say that one's from dad. This one here, we're going to say that one's from mum. This one here, we're going to say that one's from dad. So they're the 2 alleles. Now coming back here, back here, you can see HbS is 230 base pairs long, and HbA, because it's been cut, makes a 50 base pair and 174 base pairs. So I'm just going to write them down here. So I'm going to say HbA gives us a band at 50 and 174, and HbS gives us a band, sorry, I'm just going to check my thing, is 230. Okay? 230 base pairs. So now we need to apply our theory to do with gel electrophoresis and map these out on here. So they've written in your terminals, you can see negative at the top, positive at the bottom. If that wasn't there, you would have to add the positive terminal in. Always, always, always put your positive terminal down the bottom. So which one? This one here has both. So person number 1 has both of these 2 here. So that means they should have 3 bands. They should have a band at 50, a band at 174, and a band at 230. Now, you don't need to write any numbers on them. If you want to write numbers, you can. You can write 50, you can write 174, and you can write 230. Why did I put them that way around? The smaller ones are going to travel further in a given time. Now, the second person, this guy here, it contains 2 copies of this. So it doesn't have any of those short bands. So it should just have, ooh, that's a really bad, hang on, let me just read it all out. It should have 1 band at the same point as this guy did at around 230. If you want to, you can make it a little bit thicker, but there's no need to. Yeah. So your gel at the end should look down the bottom like this. So you would have got 1 mark for this one and 1 mark for that one. Have a look. Not many people got this one correct. It's a really nice opportunity to pick up some marks if you can go through and understand what we're talking about. The last, I'm going to go through another question here. There's a couple more questions to go through. So the first part of this question, and hopefully you've had a go, and if you haven't, again, please stop, have a go at the question, and then come back. So what is the appearance of a bacterial plasmid? 1 mark. What do they look like? Circular piece of what? DNA. That's it. Only 1 line. The next one is a bacterial plasmid was modified so that it contained a gene for an enzyme which contained resistance to the antibiotic. So this is what we were talking about in class, that resistance to antibiotic. Bacterial cells, which in their natural environment were sensitive to the antibiotic tetracycline, were mixed with the modified plasmid. They were then treated so that they could take up the plasmid. What is the name of the process where a bacterial cell takes up a plasmid and expresses the genes? Hopefully what you can say there is transformation. If you said bacterial transformation, that's fine too. I'm just going to pause so I can make sure that you can see my screen. This is better. All right, so what we got there was circular. You said circular piece of DNA, but the biggest thing we were looking for is the shape, which is circular, and transformation is the word that we're going here. So you could have said bacterial transformation, that's fine too, but you needed to have the word transformation. So here's the outcome of an experiment, very similar to that other question that we've gone through in class so far anyway. So the first one has got bacterial cells only spread on agar. So what we get here is lots and lots of growth. The next one is bacterial cells only spread on agar with tetracycline. So that's an antibiotic, which means it's going to kill the bacteria. And then the last one is the bacterial cells being exposed to the plasmid spread on agar with tetracycline. And what we get here are some colonies. Now there's a question mark in the middle of B there. What I look at that as I go, what would I expect? So these are untransformed bacteria on an antibiotic agar. What do we think is going to happen? Hopefully, no growth. I'm going to write that there before I even get to the question. So with respect to the growth of bacteria on plates A and C are shown. On plate A, there is a continuous growth over the surface of the agar. On plate C, you can see colonies. What result would you expect on plate B? I've already written it there. Fabulous, no growth. And explain your answer to that question. You would say all the bacteria don't contain the gene for antibiotic resistance, so therefore they die when exposed to antibiotic. And I'll bring that up on the next slide in a second when you can see it. So explain why there is a difference between plate A and plate C. So what we get between plate A and plate C is we have plate A, they're just normal bacteria with no antibiotic. Plate C, only the bacteria that have taken up the plasmid will be successfully growing there. And so what we can see is we've got that there was no growth and the bacteria are sensitive, or essentially they get killed by the antibiotic, something like that for C2. Oops, sorry. And then the last bit here says plate A, the bacteria grow because there's no antibiotic, and plate C, only the ones that have taken up the bacteria, the plasmid, can grow in the presence of tetracycline. This word here, this only, is a very important word when it comes to explaining why those colonies grow. So the last bit says it was important that students distinguish between plates A and C, recognising that the significant of the presence or the absence, either one, of the antibiotic, and also that only some of the bacteria were transformed. 1 mark was awarded for each point. Some students gave a specific percentage transformation, which could not be deduced. You didn't have enough information to say that there was a percentage there. Also, they need to be aware that if their answers include incorrect information, you won't get full marks. So even if you'd said the 2 points, but you said something that was incorrect, then they're going to take away a mark from you, which is not a great way to be. And that's the end of the questions on down to the end of page 5. Now, there are a couple of links about CRISPR. CRISPR isn't in this study design. It's actually in the next one. But if you are interested in it, please watch that video. It's one of these Kyrgyzstan, because I can't say it. One of those videos, and I will link to it, and I will put it in our playlist as well.